Beautiful. All right, you are on track and time and ready to go.
Let's share my PowerPoint then.
I should say also, Sam, real quick, so that maybe people have a bit more empathy. Sam is just coming off of a COVID-19 isolation, so he looks pretty remarkable for someone who has just gotten over COVID. So good job for recovering.
Thank you. Yeah, I'm still currently in isolation, and I don't know if you can tell with my voice. But hi, I'm Sam, and we've been kind of experimenting with using UV radiation to make these necessary quinones from these PAHs. So just a little background first. PAHs are found everywhere in the entire universe. I think it's about 20% of all carbon is found in terms of PAHs throughout the universe. They're found a lot in meteorites where they can get delivered to the Earth via, the heavy bombardment era. And then from here, we want to look at ways that it can then transform into these quinones that are important. They can be seen in the electron transport chain, used in photosynthesis and cellular respiration in terms of that. And then, we wanted to look at kind of how minerals affected this. So we took montmorillonite and kaolinite, both clays, and then we took MMS, which is the Mojave Martian simulant, which, to both explore, stuff that's found on Earth, but also can be found on Mars as well. So to start off, we looked at thermal, which Dr. Gaylor did speak about. From here, the montmorillonite, kaolinite, we were quite sigmoidal. The MMS stayed quadratic. But although we can kind of model these out and demonstrate these, once we get up to, the 500 degrees Celsius temperature, we started talking with our collaborators and some other people, and they had some concerns. We had some concerns about, is this really realistic for prebiotic Earth? Now, we can take these temperatures coming in on meteorites from outer space, coming into Earth, or certain areas on Earth itself where it's hot. But over the grand scheme, we wanted to look at a little more of an energetic source that was more useful throughout the entire planet or other planets as well. So we started looking at UV radiation as this new source of energy for this conversion. So start off, I suggested this little one-off study that was going to take just, probably 5 or 6 hours. And it ended up taking, so far it's been 6 months now. But we got these great results, this linear production as time went on, at, relatively high R squared. So from here, we saw this and we thought, wow, now we actually have to do more work for this. So from here, kind of brief experimental design, we took 1% anthracene to mineral mix. We used these glass borosilicate tubes, which we hand prepared in our lab. Then we either irradiated them in direct sunlight in the middle of summer, or used UV ones as kind of the light source in an incubator to kind of control the setting a little bit. From here, we 1 mil 1:1 DCM toluene, just kind of vortexed it to extract the produced 910 anthraquinone. Before pelleting out the mineral in a centrifuge, before using GCMS analysis for determining how much was produced. From this, we got relatively expected results. As there was more sunlight, there was more UV radiation, and therefore there was more production. We can see here that the kaolinite had by far the highest production of any of them, with conversion efficiencies all the way up into the 1.5% range, compared to the 0.3, 0.2% for montmorillonite and kaolinite. And as I said before, it was expected that it kind of happened in the afternoon that we got the highest, as you can see, through 1 to 3 p.m. and 3 to 5 p.m. And then from here, our highest average flux for a 2-hour period, which we ran it over, was 10.2 milliwatts per centimeter squared, which is a little less than what we could see perhaps in a prebiotic environment. But obviously, we do not have the UVC here on Earth that we would have without an ozone layer previously. So we were seeing very high amounts of the UVA and B instead. And then to kind of delineate how we actually got this to occur, we also took temperature measurements inside of a tube in the sunlight. And our maximum temperature was 49.3 degrees Celsius. And then when we ran these again in an incubator, in a dark incubator, all the way up to even 55 degrees Celsius, we saw no real production. So all of this was occurring based on the UV penetration of the light rather than just heating up the tube. So then from this, we wanted to kind of explore it a little more in detail. We took UVA, B, and C ones individually so that we can kind of delineate the differences between the different wavelengths and what happened. So what we did is we took samples from 30 minutes all the way up to 4 hours of these tubes inside of an incubator set at 35 degrees Celsius, just because it's a nice kind of median temperature. And then we got these kinetic curve rates for the A, B, and C for MMS, MONT, and kaolinite. As we can see for the kaolinite UVC, it's so booming that we actually had to cut off this graph, otherwise it wouldn't have fit on the PowerPoint. But what we actually saw is we saw an 8-fold increase from UVA to UVC, and then a factor of 7 times as much production in UVC compared to UVB. So as the energy of the photon is going up, we see more and more production of this anthraquinone from the anthracene. We did have to take into account a little bit of impedance from the tube itself. The UVA was impeded about 10%, UVB about 17%, and the UVC about 50% by the tube itself when we were shown the UV light through these tubes. So all this data was normalized to both the intensity of the UV wand and also the tube itself and how that attenuates the light. So then we wanted to look at, well, if we can produce this stuff, then it's great. But if it all just gets obliterated anyway, then why are we even worrying about this? Because many people tend to believe that this Martian surface, it's self-cleansing. It destroys everything organic. So there's never going to be life on there. So why are we making this stuff under the MMS or the kaolinite or the MONT if we're just going to destroy it all? So we took these samples and we replaced the anthracene with the anthraquinone. For 8 hours, we averaged out UVA, B, and C, and we saw pretty much consistent retention of the anthraquinone no matter what wavelength was used. And for all of these, we saw more than 80% remaining after virtually a day of intense sunlight. So then from here, we thought, okay, well, if we see this much, then eventually we'll obliterate it. So we went out all the way up to 3 days, and we were still seeing virtually the same amount left over, which led us to believe that, perhaps there was some small amounts of minerals on top was blocking almost all of the UV radiation that was coming in. And as a result, just a few microns to millimeters of dirt or dust is needed to kind of block all those harsh UV light that's meant to destroy all the organic matter that we see, that we could see on a different planet or on a prebiotic Earth, which is not too hard to imagine with dust storms or just simply things being moved around, and all of a sudden it's covered up by just, we're talking millimeters of dirt, where it can then react to become something that's important. So from here, we're going through some ongoing studies at the moment. We're currently looking at some more astrobiologically relevant atmospheres. All this was conducted in air. Now, although this is great, and we have about a 20% oxygen rate in the atmosphere currently, where on prebiotic Earth this was likely not the case, and definitely not the case on Mars right now. So Currently, we're exploring using different atmospheres to convert this Anthropocene to anthraquinone and see if it's still relevant. We want to look at the effects of water because obviously there's a lot of interest in prebiotic water environments. We want to perhaps look at different mechanisms for how this actually occurs so that we can understand how this would occur on a prebiotic environment, and also to look at different mineral assemblages that are also seen on Mars, maybe on an asteroid, maybe also in prebiotic Earth, to kind of create a bit bigger of a picture about all of this. So I'd like to take a moment to thank all our supporters and our collaborators and everyone who's helped us with this before fielding any questions.
All right, thanks a bunch, Sam. Good job. I appreciate that, especially since you have been very much under the weather with the stupid COVID. So we are glad that you are healing from that. So if we have questions from anybody here in the session, I don't see anything in the box here. All right. Yeah,
Mike,
so.
Can I ask a quick question?
Yep, get in there. We've got a few minutes, Jay.
Great. Hi, Samuel. I'm curious, right? Like, I'm thinking about like the weather in North Dakota. I assume there's a pretty big temperature change from like day to night, right? Like, is that true? And like, how does that play into it? Do we know? Is that known? I don't know.
We did do some studies overnight, but it was all conducted in the summertime when temperatures were still in the 10s to 20s degrees Celsius.
Okay.
The outside temperatures for this outdoor experiment was in the neighborhood of 25 to 35 degrees Celsius. We saw no production overnight though, when there was no UV, even in the kind of warmer summer nights. So it'd be likely that there would be no production kind of overnight in a colder environment if there's not even in the warmer environment.
Great, thanks.
It's a great question, Jay, and let me add to that because it's kind of a funny story. I sort of insisted that to cover the basis here for replication, real biological replication, I insisted that Sammy spend the night out collecting UV flux samples and measuring things over 3 lunar cycles, so over the course of 3 full moons, and we never saw production of anything. So it was one of those things where we were putting forth a lot of effort to get a whole lot of nothing. But we now know there's no lunar production over 3 full moon cycles here in South Dakota. So anyway.
The things we do for science.
That's right. I'm sure Sammy appreciated it. In a small town like this, he would go out and stand in the street with the UV flux meter, and I imagine you got a fair number of bizarre stares in a small town like ours. So
Definitely.
Those scientists are doing weird things again, you know. So, all right, we've got another minute or so. If there's a quick question, happy to field this for Sammy. Otherwise, we can get moving along here. All right. Thanks, Sammy. Appreciate it. Very cool.